Good morning to students. So we have completed the interface. So in interface we have studied about the three phases, that is G1 phase, S phase and G2 phase. G0 phase also we have studied. So next followed by so what happens here broadly, replication, the genetic material replicated in the interface. So then that genetic material, whatever it is duplicated, they here in M phase karyokinesis will takes place. So we have to discuss about the mitotic phase, that is nothing but the M phase. Okay, right. So what happens in the M phase? Karyokinesis, that means the splitting of the genetic material, that is the chromosomes will split, and then the genetic material, whatever it is duplicated, will be shared in different cells. So if one cell participates in mitosis, it forms two cells, exact copies. So therefore it shares the genetic material, and also followed by cytokinesis, which is nothing but the splitting of the cytoplasm. So let us discuss about the next stages, that is M phase. So keep the heading mitotic phase or M phase. So what happens in this particular phase? So here the M phase, which is equational division or homotypic division, mitotic division is mitotic phase, it is equational division. It is an equational division. So here the actual cell division occurs in this M phase. Actual cell division occurs in the M phase. Actual cell division occurs in the M phase. Next, only it lasts how many hours? I told you 1 hour, because 23 hours if you take, it happens in the karyokinesis. If you take 1 day 24 hours, 23 hours will be for karyokinesis, only 1 hour will be for the M phase. So only 1 hour it lasts in 24 hours of human cell. In human cell, it only lasts for 1 hour out of 24 hours. Okay, that is, so 23 hours is in the interphase, whereas 1 hour in the M phase. So here karyokinesis followed by cytokinesis. Karyokinesis followed by cytokinesis. Karyokinesis followed by cytokinesis. So next, number of chromosomes divide equally. Here number of chromosomes divide equally. Okay, right. Next, so this was observed by Strasburger in 1870 in the plant cells, whereas in animal cells, that is Fleming. Okay, so this was observed by Strasburger in plant cells. Okay, this was observed by Strasburger plant cells in 1870. 1870. In 1870, this was observed by Strasburg, whereas Walter Fleming in 1882 in animal cells. In animal cells, 1882. So this was the mitosis. Mitosis was observed by these different scientists. Okay, right. Next, the plant cell. In plants, it occurs in both haploid and diploid cells. In plants, mitosis occurs in both haploid it occurs in both haploid and diploid cells. It occurs in both haploid and diploid cells. Whereas if you take in the case of animal, it occurs only in diploid. In animals, it occurs only in diploid cells. In animals, this occurs only in the diploid cells. Whereas in plants, if you take, mitosis occurs in both haploid and diploid cells. Next, in social insects, it occurs in haploid cells. Exceptions you have to know. In social insects, if you take, it occurs in haploid cells. In social insects, it occurs in the haploid cells. So the mitosis can be best studied in the root apex. Mitosis best studied in root apex. In root apex, we have meristematic cells, rapidly growing meristematic cells. So therefore, here you can easily observe mitosis. Is best to observe the mitosis. Is best to observe. You can write like this. Is best to observe in root apex. Okay? Right. So, the karyokinesis, first karyokinesis. In M phase, we see karyokinesis followed by cytokinesis. So, in karyokinesis, we see prophase. What are the steps? Metaphase, anaphase, and telophase. We see prophase, metaphase, anaphase, and telophase. These are the four different phases we will study in the karyokinesis. Okay, right. So these four phases, what happens in these four phases? How does the genetic material divides? That we have to discuss. So in detail, the phases, all these phases we have to study. So keep the heading prophase. Okay. So first, prophase. So what are the events takes place in the prophase? Oh, here the changes, the nuclear changes what we observe here is condensation of the prochromosomes. Here condensation of the prochromosomes takes place. That means they are not completely formed as the chromosomes. So that chromosomes, if they condense and becomes a complete chromosome, thick and short, we call it as chromosome. Before that, the chromatin material, we call it as prochromosomes. Okay, right. So the condensation of the prochromosomes. Next, arms split longitudinally except centromere. The arms split longitudinally. Chromosomal arms split longitudinally. The chromosomal arms. You know, arms are nothing but what are the chromatids? We see two chromatids, 4 arms will be having in a chromosome. Those arms will split longitudinally except at the centromere. Except at the centromere. Okay, next. It forms 2 chromatids for each chromosome. For each chromosome, two chromatids. For each chromosome, 2 chromatids are formed. Next, nucleolus, nuclear membrane, Golgi complex, endoplasmic reticulum disappear in this prophase. So what all will disappear? Because nuclear membrane, everything, if it disappears, then only the chromosomes will come into the cytoplasm. So that will happen, sir. So here, what are the changes takes place? Nucleolus is present inside the nucleus for rRNA synthesis. So nucleolus, nuclear membrane, Golgi complex, endoplasmic reticulum, disappears. Okay. Next, the chromosomes scatter in cytoplasm. Chromosomes scatter in cytoplasm. Chromosomes scatter in the cytoplasm. So these are some of the changes. Next, the centrioles divides into 2 and moves to opposite poles. Cytoplasmic changes, what are these? All nuclear changes. Next, cytoplasmic changes, what happens means whatever the chromosomes which come into the cytoplasm, now to divide, first they have to form spindle fibers. So who is responsible in animal cells? Centrioles. If the centrioles are absent in plant cells, so then I told you already, in plant cells centrioles are absent. So who will form the nuclear spindle fibers means the cell wall itself. So remember that point. Okay. So in plants and plant cells, cell wall will form the spindle fibers. In animal cells, centrioles are present. Okay. Right. So the centrioles divides into 2 and move to opposite poles. Centrioles divides into 2 and moves to opposite poles. Next, astral rays radiate from the centrioles. The astral rays, they form. So astral rays, astral like ray-like structures radiate from centrioles. So remember all these points, events, whatever it happens in the prophase. So astral rays radiate from the centrioles. The next is initiation of assembly of mitotic spindle fibers. The assembly of mitotic spindle fibers takes place with the help of microtubules. So that is what you have to know. So the assembly of initiation of mitotic spindles takes place, I am writing here, note down, initiation of or mitotic spindle fibers. Initiation of mitotic spindles takes place with the help of microtubules, because in the last chapter only we studied what are microtubules. So centrosome has two centrioles. They are formed by the microtubules. So microtubules will form. I told you 9+2 array. So they will respond. They are 9+0 array. They are responsible for the formation of spindle fibers. Okay. So spindle fibers are initiated in the prophase itself. So all these are the events which occurs in the prophase. Okay, right. Next. So finally, if you see, centrioles are present over here. Because nuclear membrane, they are all nuclear membrane is disappeared. So chromosomes come into the cytoplasm like this. So these are centrioles. Okay. And chromosomes are scattered in the cytoplasm. So chromosomes are scattered in the cytoplasm. So this is what we see in the prophase. Okay, these are the events. The next is metaphase. So what happens in the metaphase? The second phase is metaphase. So in metaphase, whatever the chromosomes are present, those chromosomes will align at the line of equator. That means here completely nuclear membrane degrades. So whatever the nuclear membrane is present, that is completely degraded. Okay. Condensation is also completed. Condensation is also completed. Next, chromosomes are distinctly seen with two chromatids in microscope. So if you see clearly in the microscope, chromosomes are clearly seen with two chromatids. Chromosomes are distinctly, that means clearly seen with or observed with two chromatids. Okay. Next, under microscope. Under the microscope. Next, here is the best stage to study morphology of chromosome. Is the best stage to study morphology of chromosome. Best stage to study the morphology of the chromosome. So, next, spindle fibers are connected to kinetochores. The spindle fibers are connected to kinetochores. You know, kinetochores are present in the centromere. So in the centromere, kinetochores are present, two kinetochores. From each pole, the spindle fibers will attach. So we'll see diagrammatically. Okay. So the chromosomes migrate towards equator of the spindle. The chromosomes migrate towards the equator of the spindle. The chromosomes migrates towards the equator of the spindle. So all the chromosomes will migrate to the center. The centromeres lie on equator and arms are oriented towards poles. Centromere, when you see the centromere, this point is very important, location. The centromere lies on equator. The centromere lies on equator and arms are oriented towards poles. And arms are oriented towards poles. Okay, right. Okay, right. So here if you take the alignment at the equator, that is called metaphase plate. Note down. The alignment at the equator is called equatorial plate. Okay, the center, we call it as equatorial plate. One chromatid with kinetochore connects to spindle fibers. One chromatid with kinetochore connects to spindle fibers. So from one pole and its sister chromatid from opposite pole. And its sister chromatids from opposite pole. So what is this? Let us see diagrammatically how they are. So when you observe this, here when centrioles we are representing at the poles, the chromosomes will align to the line of equator. That means suppose if I draw only a big chromosome, let us see. All the chromosomes will line arrange at the line of the equator. This is called equatorial plate. At the center, equatorial plate. All the chromosomes will line at the equatorial plate. Here you see, this is centromere. We have two kinetochores. From here, spindle fiber will attach to one spindle fiber will attach to one kinetochore. From here, one spindle fiber will attach to another kinetochore. Like that. See here, that means one chromatid to one kinetochore is one part. Another chromatid with another kinetochore is another part. Like that, whatever the chromosomes are present, they will attach like this to next chromosome, to the next chromosome. They will attach like this. So this is what the arrangement is. Okay. So this kind of stage we see in the case of metaphase. All the chromosomes line arrange at the line of equator, and the spindle fibers will form from the astral rays, which are formed by the centrioles. So centrioles forms the astral rays, astrioles will develop the spindle fibers, and they will attach to the kinetochore. Okay, so this is spindle fibers, and kinetochores. So these are called kinetochores. Okay, this is kinetochore. So this is what equatorial plate. This is called metaphase plate. We also call it as metaphase plate. So this happens in the metaphase. Next, if you see anaphase. So in anaphase, what we observe, this chromatids where the spindle fibers attached, so they will pull, the spindle fibers pull these chromatid. When they pull it, chromatids will split. That means centromere will cut into two equal halves. One kinetochore with one chromatid goes to one end, another kinetochore with another chromosome goes to other end. So therefore the two chromatids get separated. Like that, how many chromosomes are there? All those will get separated. So when they are moving, the shapes of the chromosomes will come, chromatid. That is V, L, J and I, what we studied in the previous classes. So anaphase. So in anaphase, the contraction of spindle fibers occurs. Contraction. They contract. When they pull or contract, then automatically it will pull it. So centromere splits and separates two chromatids. Centromere separates, splits. What happens when they contract? Centromere splits. The centromere splits and separates two chromatids. Splits and separates two chromatids and move towards poles. and moves towards the poles. Next, that is one chromatid with one DNA. We indicate that one chromatid with one DNA. You can understand clearly. That is one chromatid with one double-stranded DNA. One DNA means it is one double-stranded DNA molecule. Because already it is splitted now, it is replicated now. So each chromatid has two double-stranded DNA. Okay? Right. So arms trail behind. The arms trail behind. Meaning of this, sir. What do you mean by this? Arm trail behind means when they are moving from the center toward the poles, when it is pulling, then the chromatid it will be like this. Whatever the arms are there, they will behind means this side. Pulling like this, arms will be trailing behind like this. So all those chromosomes, chromatids will be in this direction. So based on the position of the centromere, the shapes will be present. Okay? So that is what they are pulling. So V, L, J and I, based on position of centromere, based on the position of centromere, V, L, J and I, the shapes observed are V, L, J, I. Okay? So this happens in the anaphase. Okay? Right. So next is telophase. So finally what happens in the last phase called telophase. So this telophase is nothing but the reverse of prophase. The telophase is the reverse of prophase. What happens in the prophase? All that will be reverse in the telophase. So there everything will degenerate, here everything will form. So let us see. So in the telophase, okay, in the telophase, the is the reverse of, you can write it as, it is the reverse of prophase. It is the reverse of prophase. Decondensation occurs and lose visibility. Decondensation occurs and lose visibility. So visibility is lost as a chromosomes because of decondensation. Decondensation means again they will form into chromatin like materials. Okay. So they form chromatin network. Form chromatin network. And nuclear membrane, nucleolus, Golgi complex, endoplasmic reticulum, everything will again reappear. So in prophase they disappear, here they will reappear. Nuclear membrane, nucleolus, Golgi complex, endoplasmic reticulum, reappear. Okay. So finally, two daughter nuclei form at each pole. So finally, two daughter nuclei formed at each pole. Two daughter nuclei formed at each pole. Right. So this is what? So if you see diagrammatically how it is formed in telophase. So when it comes to one side, all the chromosomes and the pull. So here if you see at this end and this end. So we see with the chromatids are all moving to one particular thing. So these are when chromatids are moving, finally it is nothing but a chromosome. Why? Because it has one complete set of DNA molecules. So these are nucleus. So this is also another nucleus. So what we can say here, nuclear membrane, everything will again disappear. Formed. Nuclear membrane is formed. And the chromatin material, just like decondensation means, they will be in the form of chromatin material. Okay? Right. Because as they decondensed in the form of chromatin material. So next, here this is the cytoplasm, no? Cytoplasm. So two nuclei are formed in the cytoplasm. So they will split. The cytoplasm will split and divides the cell. When this splits, then it shares the cytoplasm. So when it shares the cytoplasm, automatically you will see the formation of two cells. So how does the two cells are formed after cytoplasm? Till now we studied about the karyokinesis. So here only karyokinesis takes place. So still the cytoplasm does not divide. So that phase also we have to study. So that is nothing but the cytokinesis. So when cytokinesis takes place, automatically it will divide the cytoplasm. So now if the division of the cytoplasm, the division of the cytoplasm, division of the cytoplasm, if it takes place, then we see the two daughter cells are formed in the cytokinesis. So in that also, how does the cytokinesis takes place in animal cells? It is different. The process of cytokinesis is different in the animal cells, and the process of cytokinesis is different in the plant cells. So that you have to know it. So in animal cells, we study about the cytokinesis is nothing but the division of cytoplasm into two daughter cells. So division of cytoplasm into two daughter cells. So this in animal cells, if you take, and plant cells, they differ in the cytoplasm. In animal cells, if you take, it is called cell-Furrow method. In animal cells, it is called cell-Furrow method. In plant cells, it is called cell plate method. In plant cells, it is called cell plate method. Okay. So in the next class, we will see in detail about the cell-Furrow method and the cell plate method. So then you can clearly understand how does the cytokinesis takes place and finally we have to discuss about the significance of mitosis. Okay, right. So see you in the next class. Thank you.